Very good.
Okay. So after having the introductory lecture in bacteriology, we now go to the details of some of the important foundations that are going to be helpful in your study of bacteriology. So we're going to look at bacterial anatomy and bacterial physiology. Anne Minor will be meeting in bacteriology and mycology, probably physically only during the exams due to the prevailing COVID conditions. So bacterial anatomy and bacterial physiology, and we start with bacterial anatomy. So bacteria are prokaryotes, as opposed to what you learned in physiology or anatomy, human anatomy, that human cells are eukaryotic. So prokaryotes are organisms without a membrane-bound nucleus. So at no point should we hear you talking about the bacterial nucleus. It does not exist. They do not have a membrane-bound nucleus, so they are prokaryotes. And then the protoplast, you obviously remember some of this from your study of physiology and anatomy. You must remember the protoplast. So it's bound by a thin, elastic, and semi-permeable membrane. You have already come across this. So it's a cytoplasmic membrane. It's also a phospholipid bilayer, as you learned in the human cell. Then, of course, we have a cell wall. So closely covering the cytoplasmic membrane is the cell wall. The cell wall is very important in bacteriology, as we shall see later, and as we shall see when looking at some of the drugs that target the bacterial cells. Now, not all bacteria have cell walls, as you shall learn when you're looking at the individual bacteria. Now, some bacteria also have a capsule which covers the cell wall, important for immune evasion. And of course, you know about the cytoplasm, which is an aqueous environment packed with ribosomes and other complexes that you already covered when you're looking at physiology. Now, the bacterial cells may have two basic shapes. These will be important when you're looking at bacteria under the microscope. The shape helps you to have an idea, the first idea of what you might be dealing with. So is it spherical? These are coccus, singular, cocci, plural. It could also be rod-shaped, and we refer to this as bacillus, singular, or bacilli, plural. Now, the bacillus could also have different shapes, comma-shaped bacteria. An example is a genus Vibrio, where we find the species Vibrio cholerae, which is the cause of cholera. They could be spiral. We have examples of spirillum and spirochetes that you will cover later on. And you could also have the filamentous bacilli. And to the right, you can see some of the diagrams of the different bacterial cells. So coccus, coccobacillus. So a coccobacillus, sometimes refer to them as the leiomorphic bacteria. So they are neither too large to be cocci, too small to be bacilli. So we refer to them as coccobacillus, plural coccobacilli. Vibrio, these are the comma-shaped bacilli, and of course the bacillus, spirillum, and the spirochetes that we'll cover later. So this is a cross-section of the bacterial cell. We have talked about a capsule. Not all bacteria are capsulated. So when you're looking at the individual bacteria, this is how it will be introduced. The individual bacteria will be introduced. Does it have a capsule? Does it have a cell wall? And so on and so forth. So you have the capsule, and you have the cell wall, have the plasma membrane or the cytoplasmic membrane, and you have the cytoplasm, which you're already familiar with, where you have complexes, you have ribosomes, we have plasmids, which we shall look at to see what these are. They are pili, and some bacteria are motile, and what they use is the organelle referred to as the flagellum. And we have the nucleoid. Remember, we said that bacteria are prokaryotes, so they do not have a nuclear membrane, so they do not have a nucleus. We refer to that aggregation of DNA material as a nucleoid. So bacteria have a circular DNA. So let's start with the bacterial nucleoid very briefly. So bacteria have a single circular double-stranded DNA. Most bacteria, there are some that do not have this, but most of them have a single circular double-stranded DNA, which is highly coiled so that it can fit in the very tiny bacterial cell. And the DNA, which we refer to as a chromosomal DNA, contains most of the genetic information of a bacterial cell. Not all the genetic information, as we shall see later. And it exists in a supercoiled state to be able to fit into the small bacterial cell. And some bacteria may also contain extra chromosomal DNA. And these extra chromosomal DNA are referred to as plasmids, which, as you will see later when you're looking at bacterial genetics, are important in some things like antimicrobial resistance and so on. Now, not all bacteria have plasmids. Some of them have plasmids. Then you have the ribosomes. So you already learned about ribosomes when you're looking at the eukaryotic human cell. And the bacterial ribosomes are about 10 to 20 nanometers in diameter, and they have a sedimentation coefficient of 70S. What is the sedimentation coefficient of the ribosomes for the eukaryotic cells? Anyone? Yes, 80S. So 80S, two subunits, which are the two subunits for the eukaryotic ribosomes? Yes, 60S and 40S, that's Ian, Cynthia, yes, that's correct. Now for the prokaryotes, bacteria are prokaryotes, they have two subunits. We have the 30S and the 50S subunits. Of course, they are numerous in growing cells because the function of the ribosomes is protein synthesis. And sometimes you may be able to see multiple ribosomes attached to a single messenger RNA molecules, and these are what we refer to as the polysomes. Now, the cytoplasmic membrane are similar to what you've learned in the eukaryotic human cell, thin and elastic, about 5 to 10 nanometers thick, consists mainly of phospholipids and proteins. What's the function? It's a permeability barrier, important in the transport of solids, energy generation, and creation of numerous enzyme systems. So more or less what you've already covered. Now, the cell wall of any cell is very important. As we said, not all bacteria have cell walls. Later on in bacteriology, you're going to cover some bacteria which do not have a cell wall, but most of bacteria have cell walls. Now, they are important. They encase the protoplast, and you know that the protoplast is whatever is the cell membrane and whatever is encased inside. They're also important because they give the cell rigidity, but they also have some elasticity and they are strong. Now they are openly porous to molecules less than 10 kilodalton in mass and less than 1 nanometer in diameter, and support the cytoplasmic membrane against high internal osmotic pressure. We know that with high internal osmotic pressure, if it weren't for the cell wall, then you'll have cells bursting all the time because of the very high internal pressure. On the integrity of the cell wall, and that is why we have quite a large number. Most of the antibacterial agents, as we shall learn later, target the cell wall, because once you interfere with the integrity of the cell wall, then you cause lysis of the cell wall and cell death. So we've already said this as the process of liquid disintegration and dissolution of the cell wall is referred to as lysis. Now, the cell wall also plays an important part in cell division. So in all bacteria species, the main strengthening component is referred to as the peptidoglycan. That's an important word that you'll be coming across a lot of in bacteriology, the peptidoglycan. Now, based on one of the common staining techniques, we shall look at staining techniques at the tail end of this lecture. You can divide bacteria into gram-positive or gram-negative bacteria, depending on how they appear after you carry out the gram staining technique. So the gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thin peptidoglycan layer. So what are the basic building blocks of peptidoglycan? So they have two molecules which are linked alternately. We have NAG, which is N-acetylglucosamine, and N-acetylmuramic acid. So NAG and NAM are linked alternatively to form a very strong chain. So if you have a question, you can either unmute or write it in the chat as we go along. So strands of NAG and NAM are connected by interpeptide bridges. And in addition, NAM also carries a short peptide. And the most important amino acids, or the commonest amino acids in this short peptide that NAM carries, are L-alanine diglutamic acid, mesodiaminopimelic acid in gram negatives, or L-lysine in gram positives, and then you have the D-alanyl, D-alanine. So sometimes the third amino acid may change depending on which bacterium you're dealing with, but mostly these are the commonest amino acids that are carried on NAM. So this diagram is trying to explain what we have talked about. We say the cell wall, a very important component of the bacteria that have cell walls. It gives the bacterial cell its rigidity. It protects against high osmotic pressure. And the basic building blocks are two molecules that are interconnected alternately, NAM and NAG. We've also said that NAM has a short peptide, 4 amino acids. These may change depending on the bacterium that you're talking about, but we've listed the commonest ones. So you have a chain of NAG, NAM, so on and so forth, and then you have the interlinkage. So you can see where the crosses are, the purple crosses. That's where the D-alanyl, D-alanine, is interconnecting with a different peptide from a different chain. So this makes a very strong cell wall. And to the right, you can see the basic, the peptidoglycan unit, NAG, NAM, and the short peptide. So still talking about the cell wall, as we said, the cell wall is very important in the bacterial cells. You'll be talking about it a lot. So gram-positive bacteria, remember we said have a thick peptidoglycan layer. And in addition, they also have teichoic and lipoteichoic acids. Now, gram-negative bacteria, remember we said have a thin peptidoglycan layer. And on top of the peptidoglycan layer, they have an additional layer that we refer to as the outer membrane. Now, the outer membrane is external to the peptidoglycan layer, and it contains a very important component, LPS. So you'll hear us talking about LPS a lot. LPS stands for lipopolysaccharide. Now, in some gram-negative bacteria, you'll hear us talking about lipo-oligosaccharide, which means they do not have as many molecules. So you have lipopoly, so you can have a polysaccharide or an oligosaccharide. So most have LPS, but we have some medically important bacteria that have lipo-oligosaccharide. For example, Neisseria gonorrhea, which causes a sexually transmitted infection, gonorrhea. So why talk about LPS? Why is it important in gram-negative bacteria? So LPS contains porins and anthocyanins, and this is because the outer membrane, as opposed to the peptidoglycan layer, is not freely permeable. And so any molecule that's getting into the gram-negative bacteria has to enter through some channels referred to as porins. So these are protein channels referred to as porins. And so if you have a very big molecule, It will not get inside the gram-negative bacteria. And you'll see this when you're looking at antimicrobial agents or drugs that we use against gram-negative bacteria. There are some drugs that we cannot use for infections caused by gram-negative bacteria because they're too large. So they cannot get through the porins or the porine channels to get into the target. So LPS also contains athesins. Athesins are very important. They are the initial contact in the establishment of infection. So the bacterium will adhere onto the host cell using athesins, and that establishes contact, initial steps of infection. So LPS is also important because it protects the peptidoglycan from the effects of lysozyme. Where do we find lysozyme? That you learned in physiology. In which body fluids do we find lysozyme? Lysozyme, where do we find it? So we have in tears, saliva, and milk. So that is important. This is one of the examples of natural antibacterial agents or substances. So we have lysozyme, natural antibacterial. And then it also inhibits the entry of many antibiotics. We've given an example of an antibiotic molecule being too large to go through the porine channel, and so it's ineffective against gram-negative bacteria. So we are still looking at the gram-negative cell wall. Another important component of LPS is lipid A. And lipid A forms the endotoxin. So once you start rotating in the wards, you may find patients who are in toxic shock. And the commonest cause of toxic shock is endotoxin. Endotoxin is formed by the gram-negative. So most of the patients with endotoxic shock will have infections caused by gram-negative bacteria, if it's a bacterial infection. So endotoxin may give rise to endotoxic shock when released into the bloodstream. These are extremely sick patients, and we lose most of them, unfortunately. They usually require critical care. Now, in some bacteria, you have the peptidoglycan layer being covalently linked to mycolic acids. And an example of is the genus Mycobacteria. So you're going to come into contact with the genus Mycobacterium, an important species, one of the important species in this genus is Mycobacterium tuberculosis, which causes TB. So they have mycolic acids, and so they are very hydrophobic in their cell wall. So we do not even use the normal staining techniques. They will not take up the stain because of mycolic acids in the peptidoglycan layer. So the other bullet there says mycolic acids form an extremely hydrophobic external layer, and we are staining. We need to use special staining techniques to be able to view the bacterium. So this diagram shows a cross-section of the two cell walls. We have talked about gram-negative cell walls. Gram-positives to the left have a thick peptidoglycan layer, which in that case, is brown in color. And you have the cell membrane beneath it. Remember lipoteichoic acid. The difference is that the lipoteichoic acid anchor on the membrane, while the teichoic acids are only found in the peptidoglycan layer. Then we have the gram-negatives with a thin peptidoglycan layer and an outer membrane, which is not permeable. So remember we said molecules have to get in through the porine channels. Okay, so this is very basic and this is something you need to remember throughout bacteriology. We also say that some bacteria have a capsule covering the peptidoglycan layer. Not all of them have. Most have a polysaccharide slime. There are those that have a polypeptide capsule, but most are polysaccharide capsules. Why is a capsule important? It helps to evade the host defenses and adhere onto surfaces. I'm not sure if you've done immunology yet or if it has been introduced to you, but you have different host defenses. Once a bacterium gets into the human body, then the body raises an immune response. So the very first immune response, the first responding, the innate immune system are the phagocytic cells. So they come in, they want to engulf the invading bacteria. But for the capsulated bacteria, it becomes difficult to engulf because of details that you're going to learn in immunology. But the capsule is also important because it helps to adhere to inanimate surfaces. And an example are the catheter, so either an indwelling urinary catheter or a catheter used in blood transfusion or in giving drugs, which is why when you start your rotations, you need to find out after how many days or after what duration of time should you change the indwelling urinary catheters. Because the longer they stay, the higher the chances of bacteria adhering onto this and then causing an infection. So it's also a virulence factor, so it's antiphagocytic. We've already explained that. Now, some bacteria have a polypeptide capsule. An example is Bacillus anthracis, which is a causative agent for anthrax. So we have talked about the general cell wall structures, but we have some bacteria that have alternative cell wall structures. We've talked about mycobacteria, very important medically, a causative agent of TB. One of the species is a causative agent of what you know as pulmonary TB. So mycobacteria, the peptidoglycan is intertwined with and is also covalently attached to what we refer to as the arabinogalactan polymer, surrounded by mycolic acid and a cod factor. When you're doing mycobacterium, you realize that the cod factor is a very important defense mechanism for the mycobacterium. There's also wax-D and sulfolipids. So we describe the cell wall of the mycobacterium as being acid fast. Acid fast because they are not destroyed by acids. And also the coat is responsible for virulence. We've talked about the cod factor and is also antiphagocytic. So it's able to evade the phagocytic cells that will otherwise engulf it and destroy the mycobacterium. We also have other genera of bacteria, Corynebacterium and Nocardia. They also produce mycolic acid lipids, but not as at higher a percentage as the mycobacterium. Then we have the mycoplasmas. Mycoplasma is an interesting genus of bacteria because they do not have a cell wall. So they have no peptidoglycan cell wall. So instead of the cell wall, what they do is that they incorporate steroids from the host into their membranes. So other organelles found in the bacterial cell wall are the flagella. And these, as you know, are organs of locomotion. They originate in the protoplasm and they're anchored by the basal body to the bacterial cell. So different bacteria have different arrangements of flagella. You can see to the right some of the arrangement of the flagella. So you have the monotrichous flagella. An example is Vibrio cholerae. And you have the lophotrichus, where you have tufts of flagella at both ends of the bacterial cell. You have the amphitrichus, where you have a polar flagellum at either end, and you have the peritrichus, where you have numerous flagella, the bacterial cell. These ones tend to be very motile. An example is E. coli or Escherichia coli. So other organelles, you have fimbriae and pili. Fimbriae are found peritrichously, which means they are found all around the bacterial cell, and they're more numerous than the flagella. Why are they important? They mediate adhesion between the bacterium and the host cells. And then we have the pili. So you have two kinds of pili. We have the sex pili and we have the common pili. So the common pili are also adhesins, so they're important for the attachment of bacteria onto different surfaces. And then you have the sex pili, which are similar to fimbriae but are longer. And they are so called because they initiate the process of conjugation. Of course, bacteria do not reproduce through sexual reproduction, but the sex pili are important in the sharing or the transfer of different kinds of material. For example, plasmids. Remember, we said plasmids are extra chromosomal genetic material, and these plasmids can actually be shared between different kinds of bacteria, and that is what we refer to as conjugation. So other organelles are the bacterial spores. Not all bacteria form spores. So as you're looking at the individual bacteria, it will always be mentioned. So this bacterium is spore forming or it's non-spore forming. So why are they important? Now, spores are highly resistant resting phases. So in this phase, they do not reproduce. So in their resting phase, highly resistant to different methods of disinfection and even of sterilization. So you're going to look at disinfection and sterilization later on. So these are referred to as endospores. Common genera that produce spores are Bacillus and Clostridium. And these organisms can survive in the dormant state for a very long time, and they're resistant to disinfectants. They're resistant to different methods of sterilization, drying, heating, and so on. So to make a comparison, if you want to get rid of spores, you need moist heat at 100 to 120 degrees for about 10 to 20 minutes to kill the spores. When you compare that with the vegetative cell, now the vegetative cell is the actively dividing cell. You only need about 60 degrees centigrade to be able to destroy the vegetative cell. Now the spores can remain in that state and remain viable for many years in the environment. And the process of reversal from the endospore to the vegetative cell, we refer to that reversal. We refer to it as germination. So this diagram shows a cross-section of the endospore in the middle. To the right, you have germination. To the left, you have the different arrangements of endospores in different cells. So one of the ways in which we can tell the different species or the different genera of bacteria for the spore-forming bacteria is where the endospore forms in the vegetative cell. So for some, it's at the very end, like in the second diagram. So we call those a drumstick appearance. We also have those that form in the middle of the cell and those that form almost at the end of the cell. In the middle, you have the cross-section of the endospore. So you have the cytoplasm and so on and so forth. And we said that they can survive in this state for many years until you have the correct temperature, you have nutrition, Then germination will take place and a new bacterium is going to emerge from the endospore. And then proliferation of the bacterium can then start. So remember that the endospore is dormant, so no proliferation or no division takes place in the endospore stage. Okay, so any questions on the bacterial anatomy? So if none, let's look at the bacterial physiology. So most of these are repetitions from what you've already done in physiology, but from the bacterial point of view. So you already know that physiology is the study. We look at the specific means by which a particular organism obtains energy and raw materials to sustain its growth. So you have the nutritional type. So what physical conditions does it require for growth? And these reflect the fundamental physiological characteristics. So this is not a whole subject on bacterial physiology. It is to help you understand bacteriology better. So when we tell you that this bacterium is an aerobe or it's anaerobe, what does that mean? So that when you're growing it in the lab, which conditions are you going to subject it to so you have successful growth? So the nutritional types, remember, there are three basic features used to group organisms into nutritional groups. What is the energy source? Where do they get hydrogen and where do they get carbon? So for the nutritional source, remember that energy for ATP synthesis will be obtained from light. And these are the phototrophic organisms, or through chemical oxidation, these are the chemotrophic organisms. And the hydrogen donor types, remember you can have organotrophs, which require organic sources of hydrogen, or the lithotrophs, which require inorganic sources of hydrogen. And of course, you have the carbon sources at these autotrophs. So are they able to fix carbon dioxide directly? Or are they heterotrophs, which require an organic source? And usually we combine energy and hydrogen donor designations that can be combined. So we have the chemoorganotrophs. So majority of the bacteria that you're going to look at are chemoorganotrophs. So majority of medically important organisms, but you also have some that chemolithotrophs. But what you need to remember is that most of the medically important bacteria are chemoorganotrophs. So the physical conditions required for growth, and this growth usually is growth in the lab, as we shall explain when you're talking about how we detect which bacteria is causing, for example, pneumonia in this patient. You need to grow it in the lab. So all living organisms use oxidation to transfer energy. You already know that. You know that oxidation involves the removal of hydrogen, and you also need an electron recipient. So aerobic respiration, you know that the final electron recipient is oxygen, and you have anaerobic respiration where the final electron recipient is an organic molecule. You're very familiar with this fermentation from biochemistry. So either aerobic or anaerobic respiration. So aerobic respiration, remember, results in the formation of highly toxic compounds. You have the reactive superoxide, and you also have hydrogen peroxide. So organisms that are aerobes, which means that they can survive in the presence of oxygen, produce two very important enzymes, and that's superoxide dismutase and catalase. And as the lower, you can see down there, they catalyze those reactions to give hydrogen peroxide and water, and subsequent catalase breaks down hydrogen peroxide to hydrogen and water. And so any organism missing these two important enzymes cannot survive in the presence of oxygen, because it's going to be destroyed by the formation of the superoxides and hydrogen peroxide. So keeping this in mind, bacteria are grouped according to their atmospheric requirements. So can they survive in oxygen, or do they need the complete absence of oxygen? And we said that this is because they lack the two important enzymes. So we have the strict or obligate aerobes, and these require ambient oxygen. So they're able to survive in oxygen. Oxygen is not toxic to them. And we have the strict or obligate anaerobes, and these require complete absence of oxygen. So they lack the two important enzymes, superoxide dismutase and catalase. And you also have facultative anaerobes, which grow better in oxygen, but are still able to grow in its absence. We have the aerotolerant anaerobes. So these are anaerobic, but can tolerate exposure to oxygen. We have the microaerophiles, or the microaerophilic bacteria, which require reduced oxygen level of 5%. So not the ambient oxygen of 20%. And we have the capnophiles, the capnophilic bacteria, which require increased carbon dioxide levels. Now, most of the medically important bacteria, as you'll notice when you look at the individual bacteria, are facultative anaerobes. But you also have others that are strict aerobes and strict anaerobes. So every time you're looking at specific bacteria, it will be mentioned whether these are strict aerobes, or they're facultative anaerobes, or they're microaerophiles, which is why these introductory lectures are important. So once you say these are strict aerobes, how are you going to grow it in the lab? Do you need to completely remove oxygen, or do you need to put it in ambient oxygen? So you should quickly be able to tell once you get that characteristic, general characteristic. So what about the growth temperature? So we have mesophiles. These grow in 20 to 40 degrees centigrade. We have psychrophiles, which grow best at low temperatures. We have the thermophiles, which grow best at high temperatures. So obviously, the body, what's the normal range? Normal range of the human body temperature? Yeah, so about 35 to 37, there are some just small points which you need to familiarize yourself with. So in view of that, and you can already see that most medically important bacteria will be mesophiles, because the normal body temperature falls between 20 to 40 degrees centigrade. They have to be able to survive the body temperature. Then you have the psychrophiles. These are important because if you're able to survive at low temperatures, then you can see that refrigeration will not get rid of them. Then you have the thermophiles that are able to grow at high temperatures, which is why it's important when reheating food. I think you'll deal with this when you're looking at foodborne diseases. Reheating, heating, and putting in the fridge and back. That's why you have some of these risks, psychrophiles and thermophiles. But anyway, take home for this slide is that most of the medically important bacteria are mesophiles. So bacterial growth. Why do we need to grow bacteria in the lab? So from the clinical side, you have a patient, they come with a urinary tract infection. The commonest cause of urinary tract infections are bacteria. So you need to find out which bacterium is causing this urinary tract infection. And you're going to take a sample of urine from that patient and take it to the lab. and do what we call isolation. So you want to find out which bacteria can I grow from this urine sample, or if it's pneumonia from this sputum sample, if they have diarrhea and you think it's a bacterial infection from this stool sample. So bacterial growth is important. It helps us to isolate the exact cause of the infection in the patient. So how do we know that this pneumonia is caused, for example, by Streptococcus pneumoniae? We need to isolate it and grow it in the lab. And how do we see that? So bacterial growth involves both an increase in the size of the organism because they're taking in nutrients, and also an increase in their number. So how do we in the lab then conclude that bacterial growth has happened? So it's in three main ways: development of what's referred to as colonies, transformation of a clear broth. So a broth is a liquid medium. So we have what we refer to as artificial growth media. It can be liquid or solid. So when you're using solid artificial growth medium, you're going to look for formation of colonies, which means that these bacteria are increasing, they're dividing, and you look and see colonies. But in a liquid medium, you'll not be able to see colonies. So what you see is that you had a clear fluid at the start, and now you have a turbid fluid. And this is also, if you started rotating in the wards, you'll see if you have a liquid drug, very important to always check that it's clear. If the manufacturer says that it's clear, then it must be clear. Any sort of turbidity means that there's a growth of an organism, and you could introduce bacteria or organisms into the patient as you give them those drugs or those fluids. And then the third way is through biofilm formation. Now, a biofilm, these are micro aggregations of bacteria, and this can actually be seen with the naked eye. Remember, we talked about the indwelling urinary catheters. So if they've been there for too long and a bacterium attaches to it and starts dividing, you can actually see the biofilm formation and sort of comes out as a slimy cover on the indwelling catheter. So you'll know for sure there's an infection going on, and this patient can easily get infected through bacteria getting in either the urinary tract or the bloodstream. So that's how we know that bacteria has grown. So to the left, the round, that's a petri dish. The round dish is a petri dish. You will be seeing this a lot during your bacteriology practicals. So different artificial media, we are able to identify through different ways. So for example, this is a pink medium. We already know what this medium is. And you can see these circular pink growths. So those are what we refer to as colonies. So when colonies appear, we know that you have a positive growth of bacteria. So if, for example, this was grown from a urine sample from a patient who had a urinary tract infection, then you know that this bacterium is the one causing infection. Of course, you have used quality control measures to make sure that this was not this was from the patient and not from anywhere else. Now to the right is a broth, which is liquid artificial medium. I don't know how clear it is from your side, but the one to the right is clear and the one to the left shows some turbidity. So it's not clear. So if you started off with a clear broth, then you come back the following day and find turbid broth, and you know that growth has occurred. There is some bacterial growth in that liquid medium. So what is happening as bacteria form these colonies as they become turbid? We refer to them as bacterial growth phases. So bacteria have different growth phases. We have the lag phase. So in the lag phase, there is no increase in number. The cells are preparing for growth. So they're taking in nutrients and so on. Then we have the exponential phase, which is an increase in cell number at an exponential rate until it is established the maximum available for the available conditions, because you can see we are growing them either in a tube, the broth, and the petri dish for the solid medium. And so they can only grow so far because there is limited nutrition in that tube or in that petri dish. Then you have the stationary phase, which is also referred to as a post-exponential phase. And this is where the total bacterial cell number reaches a maximum and then stabilizes. Then you have the decline phase. Now the stationary phase, so nutrition is also going down. And for the bacteria that are spore formers, this is where they start forming spores. So they don't have enough nutrition they need to survive. So they start changing and forming endospores so that they're able to survive. Then you have the decline phase, sometimes also referred to as a death phase. And this is where after a lot of proliferation, there's also release of toxic agents into the medium and some of the bacteria start dying. But the most fit and survival for the fitness. So we usually represent the growth phases in a diagram. On the x-axis, you have the time. On the y-axis, you have the log of numbers of bacteria. So you can see the lag phase. So there's no increase in numbers yet. You have the exponential phase where you have a large increase, very fast increase in the number of bacteria, but it reaches a point. So because you're either growing them in a tube or a petri dish, the nutrition is finished. So there's a stationary phase where the rate of death is equal to the rate of growth. And then you have the death phase where more bacteria are dying than are alive. And so this is where in the stationary phase, also in the death phase or decline phase, this is where you have formation of spores for those that are spore forming. So bacterial growth in the lab. So we have said we are interested in finding out this patient with meningitis, which specific bacteria is causing this. You can have an outbreak, so you can quickly say we have an outbreak of Neisseria meningitidis. We need to get in touch with the public health department so that they can start educating the public to protect themselves. So very, very important to isolate bacteria that are causing different infections in the lab. So how do we do this? We use artificial media. So artificial media, as we said, may either be liquid medium, which we refer to as broth, or it could also be a solid medium, which we refer to as agar. So the very basic medium is actually called basic medium. It has a very basic requirements for growth of bacteria. So an example that you'll be coming across is nutrient agar. Nutrient agar is solid, nutrient broth is liquid. So it supports the growth of microorganisms that do not have special nutritional requirements. They have very basic nutritional requirements. So the bacteria that are not, we call it fastidious. There are some bacteria which are fastidious. They need very exacting nutritional and environmental conditions to grow. So they will not grow in basic media. Okay, and that's the color. So once you do your practicals, when you see basic media, it's quite easy to identify. Then you have the enriched media. So we said not all bacteria will grow on basic medium. So you need to enrich it to increase your chances of isolating other bacteria. The commonest way to enrich media that we use is the use of blood, usually horse or sheep blood. So whole or lyse blood, serum, peptones, yeast extracts, vitamins, and other growth factors. So it depends on what your interest is, but the commonest enriched medium that you're going to come across will be blood agar. Very easy to recognize because it's red, like the one to the right. So during your practicals, you'll come across blood agar. Then you have enrichment media. So do not confuse it with enriched media. So enrichment media is fluid and it's selective. So selective means you use it when you're very specific. I want to isolate, for example, if you have an outbreak of cholera, I want to isolate Vibrio cholerae from this population. Do I have an outbreak of cholera in this population? So enrichment media contains substances that inhibit the growth of unwanted organisms. And an example of an enrichment medium is alkaline peptone water, which is the enrichment medium for growth of Vibrio cholerae, which is the causative agent of cholera. Now, why do you need to inhibit the growth of unwanted organisms? Now, some samples or some specimens from patients like sputum, like stool, are very rich in what referred to as normal flora. Naturally, you'll find bacteria, even in those that are not sick. So you're not interested in growing the natural bacteria. You're interested in growing the pathogenic bacterium or the bacterium that is causing disease. That is why you need to enrich the growth of the pathogenic one while inhibiting the growth of these normal flora that are not going to give you any additional information. So as you've said, an example is alkaline peptone water that we use for enrichment of stool that we suspect to be from a patient that we suspect to have cholera. Then you have selective media. Selective media is solid media. As you can see to the right, it's in a petri dish. So it's solid and contains substances. So it's selecting for a specific bacterium. So how do you select that medium to target a specific bacterium? You can add different substances, for example, biosalts, other chemicals, dyes, antibiotics, and so on. So these substances inhibit the growth of one organism to allow the growth of another organism. Again, also used when you're getting specimen from areas of the body that are very rich in normal flora, for example, stool. Again, our example here is cholera. So to the right is the medium that referred to as TCBS. So TCBS is a selective medium for the causative agent of cholera, which is Vibrio cholerae. So for example, TCBS to the right, you have seen the question, I'll answer it right now. So TCBS is usually green in color, but when you have a positive growth of Vibrio cholerae, the colonies are yellow in color. So you can see these yellow colonies. Those are the colonies of Vibrio cholerae. So the question is, what's the difference between enrichment medium and selective medium? Enrichment, the first difference is that enrichment is solid, while selective media usually is, enrichment medium is a broth, while selective medium is an agar, which means it's solid. That's the first difference. And then there's also the question is selective media the solid form of enrichment media? Yes, we have an enrichment media, which is in solid form, which I think you'll cover during your practicals, because the stool is very rich in normal flora. So you want to increase your chances of isolating the pathogenic bacterium. So you can call it as a selective medium. So you're selecting for certain things using different dyes and so on. But enrichment media, you first want to increase the numbers, because what we do if you suspect a patient has cholera, you take the stool from the patient, put it in enrichment medium. In the case of cholera, it's alkaline peptone water. And then the second day, you do what we call, you inoculate. onto the solid medium on now the selective media, which is solid. So you first increase, you might have had like 10 bacteria, so you're giving them the chance to increase in number and then transfer them to, it's called subculture, transfer them onto the selective media, in this case TCBS, so that you're able to view the colonies. So yes, you could say enrichment media is a selective form, but you are enriching the numbers of your bacterium of interest. Then when you have good enough numbers, usually in 24 hours, then you're going to subculture onto the selective medium. So you're able to see your colonies. Then you have the indicator media, which is also referred to as a differential media. So you can already see indication. So a bacterium could either give you this color or that other color, and hence the differential media. To the right is one of the commonest media that you're going to come across in bacteriology. It's a McConkey agar. So this is media to which dyes or other substances are added to differentiate microorganisms. So you can see to the right, that's McConkey agar. The natural color of McConkey agar is pink, and it contains a substance. So we usually divide bacteria into two using the McConkey agar. So it contains lactose. So some bacteria ferment lactose and others do not. So we divide bacteria on McConkey into two. So either lactose fermenters or the non-lactose fermenters, or those that do not ferment lactose. Very important when you're going to learn about bacteria that are important causes of gastrointestinal infections. So to the right is the same petri dish divided into two. So you have two different bacteria growing there. So to the left, you have those that ferment lactose. Usually they'll give you pink colonies. And to the right are those that do not ferment lactose. So those ones will give you the pale or the yellow colonies. So a very common example of a bacterium that ferments lactose is E. coli or Escherichia coli. I'm sure you've heard of E. coli. A good example of one that does not ferment lactose is Proteus, a very important cause of urinary tract infections. So indicator differential media divide bacteria into two. Does it ferment lactose or doesn't it ferment lactose in the case of McConkey agar? But you can have others that you will come across when you're looking at urinary tract infection. So we'll not go into the details now. Then you have transport media. Why is it called this? So you are working in a place without a laboratory, and you need to isolate the bacterium that's causing an infection. So how do you make sure that the integrity and the viability of the bacterium is maintained until you transport the bacterium to Nairobi or to the referral hospital? So we use transport media. Now, transport media tends to be semi-solid. To the right, you have Stewart's medium. So it tends to be semi-solid, and it contains ingredients to prevent the overgrowth of commensals, normal flora. Again, normal flora can give you wrong results. They overgrow, so they're not helpful because these are normal flora. They're unlikely to be causing the infection. You're interested in the pathogenic bacteria. So to the right is what refers to as Stewart's media. Stewart's media is an important transport media when you are looking at diseases, let's say sexually transmitted infections. A common one is gonorrhea. So these bacteria tend to be very fastidious. They're not going to eat 24 hours. You'll find that they've died, so they'll not grow in the lab. So you need to maintain the integrity and their viability. So to the right, you have the tube containing the semi-solid media. You can see it at the bottom of the tube. There is some creamish substance, that's the Stewart's medium. and then you have a swab. So what you do is that you take a swab. This is a large one, so this is probably from, obviously this cannot be for a penile swab. This is for a vaginal swab or an endocervical swab. So you take the swab immediately, put it inside the tube, and you make sure that the tip of the swab that contains what you have swabbed from the patient is inside the transport media, and then you can transport it to the lab. And different manufacturers will tell you, so viability is maintained for 24 hours or 48 hours or 72 hours. So you'll have enough time to transport it to the lab, and then there you're going to transfer this onto the solid media, and then grow it in the lab and get the colonies and make a conclusion about what is causing the infection. So important because it ensures the survival of aerobic or anaerobic. It depends on what you're investigating when specimen cannot be cultured immediately after collection. So an example, as you see this, Stewart's media. Then we have the identification media. So the flow of work in a bacteriology lab, you have a specimen from the patient. You do isolation. So we've looked at what we refer to as primary isolation. We have seen some of the colonies that, for example, Vibrio cholerae will produce, but you still need to identify. So you have an idea that this is a Vibrio, but is it the species of interest? You need to go further and do what we refer to as identification of what you have isolated. So there are different ways of identifying what is growing on your artificial media. So we have identification media and so on and so forth. So identification media to the right, you have different reactions to the same medium. So the original medium was actually the fourth tube to the right, and you can see the different reactions that you can get with different bacteria. So we'll not go into the details now. You're going to look at this when you're looking at the different bacteria. But from the left, the yellow reaction, and you can see there's some space at the bottom of the tube. That means that this reaction of this bacteria resulted in some gas production, which is why there's some space in the middle, and the agar is raised. And you can see the different colors. So when you're looking at the different bacteria, you'll be taught how to tell which bacterium gives you this yellow reaction, which bacterium gives you this yellow and pink, which bacterium produces hydrogen sulfide, because this black color shows that there's production of hydrogen sulfide gas. So this is referred to as a triple sugar ion, TSI. We'll not go into the details. You're going to learn a lot about it when you're looking at the enteric bacteria. So organisms are identified by changing color of the medium. You can see the different color spectra and or production of gas. So when there's a raising of the medium from the tube, there is gas production, hence the space between the tube and the tip of the medium. But the details will come later when you look at the specific bacteria. So what about the growth atmosphere in the lab? You must be able to mimic the natural needs of the different bacteria. So if you're dealing with Vibrio cholerae, can it survive in ambient oxygen? Does it need complete absence of oxygen? And so on and so forth. So aerobes and facultative anaerobes are cultured in air, which means you do not need any special equipment because you already have the oxygen in the air. But you have that special category of bacteria, the strict anaerobes, which require complete absence of air. Remember, they do not have the two important enzymes, so oxygen is toxic to them. So either an anaerobic jar, which means that you take off, use mechanisms to completely remove oxygen, or what's referred to as a field Macintosh jar. So that's another name for the anaerobic jar. Some labs have anaerobic incubators. So we're going to talk about incubators. So for capnophiles and microaerophiles, additional carbon dioxide, either using a candle extinction jar or a carbon dioxide incubator. Now, temperature, remember, most of the medically important bacteria are mesophiles. And remember, room temperature is about 25 degrees centigrade. So you need to achieve 35 to 37 degrees centigrade. And you use that, and you use an incubator to achieve that temperature. So to the left, a Fields Macintosh jar for anaerobes. To the right, a candle extinction jar for the microaerophiles and the capnophiles. So this usually you understand it better when you do the practicals on it. So light a candle, it's going to use up all the oxygen there, but not all the oxygen. So these are not strict anaerobes. They just require an additional percentage of carbon dioxide. But for Fields Macintosh jar, also referred to as the anaerobic jar, you totally take out the oxygen from there to assure the growth of the strict anaerobes. Okay, so this will become clearer once you do the practicals in the lab. So any questions of bacterial anatomy or physiology? So any questions at all? So are errors common in culture methods? So Paul, maybe you could ask, what do you mean? Do you mean, for example, if you have an aerobe, then you put it as an anaerobe? Is that what you mean? What you'll come to realize when you do bacteriology is that, and you've been told this, I think, many times, common things will occur more commonly, but that doesn't mean that you can not have the uncommon things. So if a patient has meningitis and you are in Kenya, and it's a bacterial meningitis, they are the commoner causes of meningitis, Neisseria meningitidis. So you already know that this is a facultative anaerobe. It's rare. In fact, I'm not sure of any that, at least in the adult population, is a strict anaerobe. So as you learn bacteriology, it will become easier once you learn the individual bacteria. So this bacterium is called Neisseria gonorrhea. It causes gonorrhea. So you already know this is how it presents. And so when you're going to culture in the lab, you already know this is a capnophile. You need to grow it in a candle extinction jar. But when you're not sure, because you don't know what's causing this meningitis, so you have the commonest causes of meningitis. Those usually come from research. So, you know, for example, in KNH, meningitis is mostly caused by this, or if in this, there's an outbreak, so you're most likely to be dealing with this. So you know what environmental conditions to expose the bacterium to. Or what errors are you interested in? Do you mean errors of wrong temperature or wrong medium? Or is that clear?
Yes, it is clear.
Okay, any other questions? So this may seem a bit abstract because it's a lecture, but once you do the practicals, it will become easier, quote unquote, once you start doing the individual bacteria. So this is facultative anaerobe. It does not form spores and so on and so forth. And then when you're doing the systemic bacteriology, where you look at meningitis, what are the commonest causes of meningitis, what are the commonest causes of pneumonia, then you use the knowledge you learned from the individual bacteria to guide you in isolation of the bacterium in the ward, in the lab. And that is why for the laboratory request forms, you need to have a sort of history. If it's blank, then we use too many resources to be able to isolate the bacterium. But if you tell us it's a 1-day-old baby presenting with meningism, then you already know the common causes of meningitis in a newborn. So you're more likely to use this kind of medium, expose them to this temperature, expose them to this, whether is it anaerobe, strict. That's why our history is very, very important when you're taking your specimen to the lab, so that we don't use too many resources and too much time trying to figure out, and too much back and forth trying to figure out, is this Streptococcus pneumoniae, is it Neisseria meningitidis, is it likely to be, not is it, of course it could be different, it could be a rare cause, but you have the commonest causes. Any other questions? Okay, so if no questions, in summary, we have looked at the bacterial cell structure, important in your lectures in bacteriology. We have looked very briefly at the physiology. You already know physiology, so this was from a bacteriology point of view. So this is to help you in the upcoming lectures in bacteriology. Okay, so if no other questions, then we meet in January.
Thank you.
Okay.
Thank you, Doc.